Welcome to this explainer. Today, we're doing something pretty cool. We are going to shrink down and explore the microscopic anatomy of the trillions of organisms that are either keeping you alive right now or trying to make you critically ill. You know, every single second of your life, you are playing host to microscopic worlds. Some of these bacteria are great. They help you digest food. But others, they can cause devastating infections, things like tuberculosis, cholera, and sepsis. To truly understand how they do this, we've got to look at how they're built. These little guys are so small they typically measure just 0.5 to 5 micrometers across.
Now, if there is an ultimate cheat code to microbiology, it's absolutely this. Understanding clinical medicine requires mastering bacterial anatomy. Seriously, how do antibiotics kill bacteria without harming your own cells? Or how does a lab test know exactly which bug is making you sick? Well, the answers to all of these questions lie directly in the structural components of the bacterial cell.
So, here's our game plan for today. First, the bacterial blueprint. Then, the outer defenses. Third, movement and attachment. Fourth, the inner machinery. And we'll wrap it all up with some high-yield exam pearls.
All right, part 1, the bacterial blueprint.
So, right off the bat, we need to establish that bacteria are vastly different from our own human cells. As prokaryotes, they completely lack a true nucleus, and they have zero membrane-bound organelles. Plus, while human DNA is organized into these nice linear chromosomes, bacteria just carry their genetic instructions on circular DNA, and they multiply through a simple process called binary fission. Now, why does this matter? Well, these foundational differences are exactly why we can design medicines that specifically target them while leaving us completely unharmed.
Moving right along to part 2, the outer defenses.
Picture a microscopic force field around the cell. That's the capsule. It's this outermost gelatinous layer. And while they're usually made of polysaccharides, there are always exceptions, right? Like the capsule of Bacillus anthracis, which is uniquely made of poly-D-glutamate. But what does it do? Well, not only does this capsule prevent the bug from drying out, it makes a bacterium incredibly invasive. It literally shields it from our neutrophils and macrophages, stopping our immune cells from swallowing the bacteria whole, a process known as phagocytosis. It also acts kind of like glue, helping bacteria firmly adhere to tissues and form sticky biofilms on medical devices. Because these capsules are such a massive virulence factor and they're highly antigenic, their polysaccharides are actually major targets for vaccines. If you're prepping for medical boards, you will absolutely see this on your exams, for sure. So commit this SHNISKS mnemonic to memory. We're looking at the heavy hitters here: Streptococcus pneumoniae, Haemophilus influenzae type B, Neisseria meningitidis, E. coli K1, Salmonella typhi, Klebsiella, and Group E strep. Knowing exactly which bugs wear this heavy armor is essential for clinical practice. Okay, sitting just beneath that capsule is the cell wall. This gives the bacterium its shape and provides some serious rigid osmotic protection against lysis. It's built out of this unique material called peptidoglycan or murane. Think of it like a chain-link fence made of alternating sugar steps, N-acetylglucosamine or NAG, and N-acetylmuramic acid or NAM. These steps are securely cross-linked by peptide bridges. And get this, those vital bridges are built by enzymes literally called penicillin-binding proteins. Keep that in your back pocket. Now, depending on how that wall is structured, bacteria basically fall into two main camps. And honestly, this structural dichotomy is probably the most important concept in all of bacteriology. Gram-positive bacteria, they've got a massive, thick peptidoglycan layer packed with teichoic acids, and they turn a deep purple under a gram stain. But gram-negative bacteria, oh, they're sneaky. They have a really thin peptidoglycan layer hiding beneath a complex outer membrane. That membrane contains a periplasmic space and transport channels called porins. Crucially, it also contains lipopolysaccharide. This has a highly toxic component called lipid A or endotoxin. Let me tell you, the presence of lipid A in gram-negative bacteria drastically changes their clinical impact, frequently triggering fever, hypotension, and even full-blown septic shock. Remember earlier when we talked about exploiting the structural differences between human and bacterial cells? Well, because human cells completely lack a cell wall, we can deploy powerful antibiotics, like penicillin, cephalosporins, vancomycin, and carbapenems, to safely burst the bacteria without doing a bit of damage to our own tissues. These drugs essentially jam up the machinery that builds those peptide bridges in the peptidoglycan layer we just talked about, completely destroying the integrity of the cell wall. But hey, microbiology loves its exceptions, so consider this tricky little scenario. What happens when typical cell wall inhibitors like penicillin are completely useless? Which bacterium breaks all the rules by lacking a cell wall entirely and actually steals hosts' cholesterol? Any guesses? Yep, you guessed it. It's Mycoplasma pneumonia.
This guy survives without a cell wall entirely. To keep its cell membrane stable, it uniquely incorporates cholesterol directly from the human host into its own cell membrane. So if a patient comes in with a mycoplasma infection, hitting them with a drug that targets a cell wall that simply doesn't exist, yeah, that's not going to get you very far.
All right, moving into part 3, movement and attachment. So how do these tiny things navigate our bodies? Flagella. Made of the protein flagellin, these are essentially microscopic outboard motors that dictate motility and chemotaxis. Basically, it's how bacteria intentionally swim toward food or run away from danger. And they actually come in several distinct arrangements around the cell. You have monotrophous bacteria with just a single propeller, like Vibrio cholerae. Then you've got lophotrophous, amphitrophous, and finally peritrophous bacteria, which are literally covered in them. Think E. coli or Salmonella. Oh, and a quick lab tip. We actually use this flagella protein, known as the H antigen, to identify specific bacterial strains.
Now, once they reach their destination, they need to stick around. This is where fimbriae and pili come in. They use short, highly numerous fimbriae, almost like microscopic Velcro, to firmly stick to human tissues and colonize our organs. On the flip side, they utilize the longer, much less numerous sex pili to actually connect with other bacteria and swap genetic material in a process called conjugation.
Which brings us to part 4, the inner machinery. Floating deep in the cytoplasm is this magic number, 70S. This specific number represents the bacterial ribosome, which is the protein factory of the cell. In the weird math of microbiology, a 30S subunit plus a 50S subunit equals a 70S ribosome. Now, since human ribosomes are a larger 80S, this makes the bacterial 70S ribosome a perfect, selective target for crucial drugs like gentamicin and azithromycin. Also hanging out inside the cytoplasm, you'll find plasmids. These are small, extra rings of DNA that replicate completely independently of the main bacterial chromosome. You can think of these plasmids as genetic power-ups that carry codes for dangerous toxins and multi-drug resistance. Bacteria actively trade these power-ups with each other via their sex pili to rapidly spread antibiotic resistance across different populations. It's literally a microscopic black market of survival traits.
And what happens when environments turn completely deadly? Extreme heat, radiation, chemical disinfectants? Well, certain bacteria have an ultimate doomsday protocol. These specific gram-positive genera survive by forming incredibly resilient, non-reproductive endospores. Just remember the mnemonic BACS for Bacillus and Clostridium species. We are talking about the heavy hitters that cause anthrax, tetanus, botulism, and C. diff. These spores can lie dormant for years, which is exactly why rigorous medical sterilization is absolutely vital.
Okay, let's pull all of this together in part 5, high-yield exam pearls.
Let's rapidly review how anatomical structure perfectly links to clinical function. The capsule, that's your antiphagocytic virulence factor. Peptidoglycan provides cell wall structure through alternating NAG and NAM sugars. Lipid A, that triggers endotoxic shock. Ribosomes are your 70S antibiotic target. Flagella enable motility and represent the H antigen. Pili facilitate genetic conjugation. And endospores, they guarantee environmental survival for Bacillus and Clostridium species. Seriously, study these exact pairings. They are the absolute core of medical microbiology.
Because look, memorizing bacterial anatomy isn't just some academic hoop to jump through. It is the very foundation of how we diagnose, treat, and defeat infectious diseases on the ward. Without understanding the cell wall, we don't understand penicillin. Without understanding ribosomes, we don't understand azithromycin. Every single piece of this tiny machine tells us exactly how to break it.
So, I'll leave you with this final thought to chew on. As our current anatomical targets begin to fail due to plasmid-driven antibiotic resistance spreading across the globe, where should the future of microbiology look next? How do we outsmart a microscopic machine that is constantly rewriting its own blueprint? Keep exploring, keep questioning, and I'll see you in the next explainer.